I thought as the first speaker, and as a vascular biologist and a medical doctor, my area is internal medicine, so I think about the whole body, but have spent many years looking at cancer. I thought I would really focus on rationale in terms of validating the idea of dietary cancer control, which is along the theme of developing evidence. And I'm going to talk about sort of the big picture of diet and cancer, and I will bring it to my own experience in terms of how evidence is currently being developed. And when I say currently being, what I'm saying is that we are still very much learning how to actually develop this evidence. And so as it accumulates, we can put it together, integrate evidence together as we will during this day, and hopefully have more scientific and clinical collaborations that will allow the field to stand on its own and not be viewed as sort of a side field that is not mainstream, but in fact to bring it into the mainstream. As Bernard has alluded to, my work in angiogenesis, which led to our introduction many years ago, this was angiogenesis itself was a side field, but it really took a focused scientific approach married to a clinical approach in order to be able to bring validation to it. So with that, I'll start with this infographic that shows different causes of death in the world. And of course, the odds of dying from any cause is 100%. We will all go for some reason. And when you look at many different types of reasons, like drinking too much alcohol, or drowning in the swimming pool, or perhaps having an automobile accident, which is tragic and common, you can see that cancer, in fact, trumps many of those other causes, and in fact is only second to cardiovascular disease as a common cause of death among many different ways that we will all go. This is perhaps why cancer research is an excellent focal point for examining new fields, such as the impact of diet and health. And what I'm looking at the program and imagining is that many of the scientific concepts and even clinical implications that we'll discuss today not only apply to cancer, but may apply to other diseases of life, including chronic diseases, but potentially, perhaps even more importantly, give us some insights into how to remain healthy and how to protect our health early on. The other sort of importance of cancer, of course, is its scale on a global level. If you look at numbers from 2010 to projected to 2030, this is the new data from the AACR showing that the growth from 13 million cancer cases to 21 million, this doubling is going to occur if we don't actually try to prevent the disease. And smoking cessation is one of the important interventions, but there are many others, and people approximate perhaps up to a third of cancers may be associated with diet, and oftentimes the association is what is the negative element in our diet that we should avoid to avoid cancer. But what we're here to talk about today is, are there other ways to view diet, including fasting or metabolic control, or the intake, consumption of certain dietary bioactives, which I'll talk about. The other issue is economic. Cancer is a very costly disease. It's costly in direct costs of treatment. It's also very costly in terms of indirect costs of loss of income and productivity for the countries. You can see that if you project from the $290 billion of cancer costs from 2010 to almost $50 billion in 2030, this cost will exceed the current GDP of these 5 countries, Austria, Norway, Denmark, South Africa, and Ireland. So it's quite stunning to think about the cost. Now, much of the cost is involved with treatment, and the new treatments for cancer, which I will also mention briefly, are very exciting but also very expensive. And so the other implication of diet is that there's maybe an opportunity to help not only save costs through disease prevention or cancer prevention, but also potentially an adjunctive treatment using our diet that is largely ignored by the oncology community, really because of lack of data and lack of education, to be able to achieve clinical goals that we would like to have. So the topic of validation is not one that's only for diet. In fact, from the very beginning, in the history of oncology, surgery had to be validated, chemotherapy had to be validated, and radiation had to be validated. And these are all the mainstays for cancer treatment. And while they often fall short from what we would like to achieve with patients, they can also be quite effective in some cases. And in many cases, they allow the patients to live longer from the time of diagnosis and represent the best of practice that exists in many places around the world. Now, this has changed over the last 15 years, maybe almost 20 years now. Targeted therapies worked through very difficult beginnings with just discovering what are some targets on the cancer cell, in the microenvironment of the cancer, in the matrix, in the vasculature, and now in the immune system, which has led in kidney cancer as a beginning, but many other cancers now, lung cancer, endometrial cancer, melanoma, to give us an understanding of how our own body's defenses through the immune system may wind up becoming much more powerful than we ever imagined, number one, and number two, perhaps more durable than our other treatments at hand, like chemotherapy. And so I just want to show you some of this. A human face always helps to make the case. This is my colleague Stephanie Joho. She's 28 years old. Her mother died of Lynch syndrome, which is an inherited genetic time bomb for multiple malignancies, and she was in college when she had bleeding in her stool and was went to the emergency room and diagnosed ultimately with stage 3 colorectal cancer. And at the time, she went to very good oncologists in New York City and sought treatment, which was involving surgery and chemotherapy. They considered some radiation, and her cancer continued to spread. And she was actually given a palliative approach with just going home with pain management. Her cancer was essentially unstoppable using the traditional, conventional means. And her younger sister, as so often happens today, works in the media and was very media savvy, went on the internet, and found a clinical trial at Johns Hopkins University on immunotherapy. And by looking at the molecular profile of her colon cancer, they found a marker which suggested that she would respond to an immune therapy, a checkpoint inhibitor, and indeed, within a few months of treatment, in fact, all of her tumors disappeared. And so this is what she looks like today. This has now led to, among other work, an FDA approval on the molecular signature for microsatellite instability. She's been in the media, sort of like JJ has, as a champion of trying to move progress forward from the patient perspective. There's also a companion to these new treatments, which is to do molecular profiling in real time. I'm going to come back to this at the very end, but you can look at the entire human genome in the tumor, compare it to normal leukocytes and compare that genome, look at the transcriptome, look at the proteome, use artificial intelligence to compare, look at what the targets are, and then select treatments in a precision fashion. And there's also now liquid biopsies allowing you to use blood to look for circulating tumor DNA and RNA. We're at the very beginning of this, but I'm already involved with some of this type of work. Now, angiogenesis, which is my area, describes how the body grows blood vessels, and tumors are able to hijack that process of growing blood vessels in order to be able to feed themselves with oxygen, nutrients, growth factors, and other survival factors. And so within the environment of the cancer, blood vessels become one of the actors that actually help to enable the cancer to grow from a small, essentially harmless mass, as you can see on the left, because we all are forming cancers in our bodies continuously, even now. And then when they are able to grow a vasculature, they can actually trigger explosive exponential growth. In the laboratory, once blood vessels touch a tumor, the cancer can grow in volume 16,000 times in just 2 weeks. So the idea is, can we actually cut off the blood supply to tumors? This is something that was pioneered by my mentor, Judah Folkman, and then subsequently more than 1,000, now 10,000 researchers and clinicians have entered the field. And it's led to a number of anti-angiogenic agents that have been FDA approved starting in 2004, as you can see on the top, first for colorectal cancer and then for other types of cancers. And now there's 20 different human anti-angiogenic cancer treatments that belong to targeted therapy. There's even one in the middle, which is in 2009, toceranib, which went from humans to animals. The veterinary world actually borrowed from the tyrosine kinase inhibitors for angiogenesis for canine mast cell tumors. And so this is where we can learn from our animal companions, not only in the laboratory as mice, but also to try to give back to the animal world. And this field has been able to cross-fertilize that way. So to move to our topic, what about validating dietary control of cancer? It's not a new idea. In fact, if you look in other cultures, Asia, many countries in Europe, in Germany, and including in France, the tradition of diet in health and diet in cancer goes back a long time. But it has, until recently, not had the scientific foundation and the integration that I'm going to show you now and we're going to hear about today. So the first thing as a scientist, as a vascular biologist, I ask in terms of validation are some basic questions. So first, does it work? How does it work? How do we apply it if we want to actually use it for treatment, whether it's in the laboratory or in the clinic? And then how do we monitor the results? And I think that if we can begin to ask these questions in responsible and rigorous ways, then we can establish the basis for really using and diet methods as a mainstream, as a companion in everything else that we have for cancer. So it's challenging to validate dietary cancer control. And you know, many of us who work in and have experience in the biopharmaceutical area, including here at the Institut Gustave Roussy, is we always think about developing evidence as randomized, double-blind, placebo-controlled trials. Very difficult to do that with food. There are some small studies that attempt that, but then they are small studies. It's a very difficult in a large population way to actually tell people only to eat tomatoes or potatoes or certain vegetables for long periods of time that we may need in order to see these outcomes. And so I think finding this level 1 evidence, which has become one of the gold standards of medicine, is something that we all need to aim for, but recognize how difficult it is to do here. Now, the other way to do this, besides a pharmaceutical style trial, is really to look at compiling the different types of evidence that exist at the genomic level, molecular level, in vitro or cellular level, looking at animal data, clinical studies, which tend to be small, 10 to 100, well-designed, controlled studies have been done in foods. And then there's population studies, and here in Europe, the EPIC trial, the EPIC study is really indeed one of the most important collections of data that is continuing to give us insights on how dietary patterns associate with outcomes, including cancer. So moving to how things work, in looking at this program, I think these are the things that we are going to touch on and perhaps more. I'm going to talk about angiogenesis in just one moment, but we can alter cancer cell and host metabolism. We can enhance and alter and control, modulate the immune response. I'm not going to talk about that, but I'm also involved with looking at dietary control of immunity. There's also influence on DNA repair, influence on the microbiome, which I'm also not going to have time to talk about. I'll touch on briefly. So there's many mechanisms of how diet can actually work, and these mechanisms communicate with one another. So the microbiome, the diet goes into the gut, it connects with the immune system that lines the gut. The microbiome or the microbiota communicate with the immune system. The immune system communicates with other parts of the body. So this is a systems biology enterprise that we are entering, and not a reductionist one. And I know there are some journalists here who have been very enthusiastic in this topic. And one of the challenges, I think, in communicating this field to the public is not to oversimplify and yet still give people something that they can actually understand. There's something that they can do on a pragmatic level. So I gave a TED talk on angiogenesis that was called "Can We Eat to Starve Cancer?" introducing the idea that what has been accomplished in biotechnology could be applied to study diet. In other words, the methodologies, the research tools could be converted from looking at drug development, and you can crank the turret and refocus the lens on food and some of the remarkable things that we've discovered over the years. This has been quite a popular TED Talk. Some of you I know have seen it. The last count is 11 million views, and so there's a big appetite for the topic. And what I was describing, and I'm going to update you now on what we're doing, is really beginning to take a look at whole foods that one would find in the market, and begin examining what Mother Nature has actually imbued the foods with through evolution. Now, most of the bioactive chemicals found in whole foods, especially plant-based foods, evolved from over the genuine evolution, really to provide protection or defense to plants and their fruits. So they either provided protection against pests as natural chemical insecticides, so many of the bioactives have that natural function for the plant, or also they help to attract insects for pollination. So they helped reproductive defense to protect the species. And so when man co-evolved with plants and began eating the plants, those natural chemicals began having a new job description, new function, which is that they were swallowed by humans, and they came in contact with human cells, and then a third job description became there, which is to interact with human tissues. And this is really what I call an undiscovered country. There is an enormous amount of information that we have yet to learn about what the natural chemicals in plants that serve the biological function in plants, what they do in the human body. So dietary anti-angiogenesis is a corollary to pharmaceutical anti-angiogenesis, and it goes even further, because at the very end I'll tell you some of the work, a bit about some of the work we're doing looking at the other side of the equation. This is a list of different sources of dietary anti-angiogenic foods that we've actually found. I want to just show you how evidence has developed over time by taking a plant, the soybean, a plant-based food, and show you how this evidence is developed. So everybody has had probably an edamame in a Japanese restaurant. It's a very important, actually, foundational food for Western culture as well as Eastern culture. But this, in 1991, was the pivotal paper that came out of the University of Helsinki, including a Greek researcher, Ted Fotsis, who inherited urine samples in a laboratory from a prior researcher who studied steroids in women and was looking at them in women. And so there had been, from a previous collaboration, frozen urine from a Japanese village, from villagers from a Japanese village outside of Kyoto, where the idea was to perhaps study their steroid content. And what this group did is they compared what kinds of foods that these subjects ate. There were 17 of them, and they were all farmers, men and women. They ate what they grew. It's a rural village outside of Kyoto. And then they looked in their urine and found discrete peaks that they could actually identify, one of which was genistein. And genistein is a phytoestrogen, which subsequently was discovered to be a dietary inhibitor of angiogenesis in vitro. And we've taken this on by working with the U.S. National Cancer Institute in a model in which we're studying angiogenesis in these rings, of aortic rings, that actually are lined with endothelial cells, which grow blood, which are what blood vessels consist of. And you can stimulate the system to have this outgrowth of blood vessels, a starburst. And when you put genistein, derived from soy, into the system, you can see a very potent inhibition of angiogenesis, similar to the way that tumors would grow blood vessels, and that anti-angiogenic drugs would stop those blood vessels from growing. This is what the effect of genistein is, and it's consistent and builds upon Ted Fosis's work. Now, subsequently, other groups, including this group in Taiwan, began looking at the molecular mechanisms of action, and they found many. they found that genistein inhibits in endothelial cells growth factors like VEGF, PDGF, urokinase, plasminogen activator, proteases like MMP2 and 9, and upregulate endogenous inhibitors of angiogenesis like angiostatin and endostatin and the thrombospondin. So for those of you in the dietary world, this may be alphabet soup, but for those of you who are from the Institut Gustave Roussy who have worked for many years in this area, you'll know that these are very highly valid and important targets. So this is the way that we look at this at the Angiogenesis Foundation, is to look at the entire spectrum of blood vessel growth and say, where do those targets hit? And in the X's is where the targets are hitting from soy, from blood vessel growth. And also in the box on the far right in red is where the natural inhibitors are coming up. So this is actually shifting the microenvironment towards an anti-angiogenic outcome. Now, is there any human evidence or patient translation of this data, any association? Because what you do in the lab often fails to bear fruit in people. And so this is where you jump to epidemiology. And I'm going to beg forgiveness from my colleagues associated with WHO, because I'm not going to be able to speak with the same authority about epidemiological studies. But this is a publication from 2009 examining in Asian American women childhood, adolescent, or adult soy intake, and finding that, in fact, soy intake early in life is associated with decreased incidence of breast cancer, decreased risk. And this is from 1,563 patients, so it's actually quite a large study. Now, it's a risk, and it's an association of decreased risk. What if you actually have breast cancer? So this is a huge study, 5,000 patient study, this Shanghai Breast Cancer Survival Study, in which they looked at women in China who have breast cancer and what their association is between soy intake and cancer outcome. And it showed that women who actually ate soy had up to a 29% reduction in the risk of death and a reduction in recurrence by 32%, with the association of about 10 grams of soy per day. So how much is 10 grams of soy? It's about a cup of soy milk, or 1 cup of edamame, which you can have in a restaurant anywhere, or tempeh, which is Indonesian, or soybeans. So not really unreasonable amounts is where this association comes from. Now, I just told you Asian American women, and also for risk, and also a study in Shanghai. So is this effect somehow genotypically in the Asian population, which is a very different environment? Well, this is a study in North America looking at the breast cancer family registry involving many different ethnic types, looking at the same thing. This is 6,842 patients, and finding that in an ethnically diverse cohort, the higher intake of soy reduced all-cause mortality, including cancer. So this includes Caucasians, Hispanics, African Americans, and also Asians. So we've actually been looking at this type of string of evidence, from molecular to animal to cellular to animal to public health, and beginning to see, can we actually do comparative activity? And you can see soy we've studied here, and we actually compare the activity in the lab to chemotherapy and other targeted cancer drugs, and you can see it's a very interesting way to look at the opportunity to look at food activity. Green tea is another area that I think you may be familiar with, many catechins in green tea. I'm showing you this really not to just keep on talking about another food, but to show you the methodology. This is a fertilized chicken egg growing in a dish, also used to study angiogenesis, and if you can actually put onto the vascular growing network tea or any other food substance, and you can see on the right that when there's an anti-angiogenic substance in the natural compound, the blood vessels are actually dissipate. And you can also use an implant in the cornea. Here in a rabbit, this is a mouse, where you can give mice tea in drinking water and implant a substance that will grow blood vessels around the ring of the cornea. You can see there, they're growing up towards the implant, which acts like a tumor. And then if you just add drinking water, you can then quantify the effect from dietary intake of tea, and you can see much fewer blood vessels there. This can be counted, you can do the histology, you can look at the mechanisms of action. We can then actually take it further to look at the differences between different tea types, and we looked at the differential activity between jasmine tea, sencha tea, Earl Grey tea, and then some blending. And what we found was really interesting in an in vitro assay, which is that combining the teas could actually lead to some synergy. So one tea plus one tea led to even a greater effect than the summation of them alone. So again, is there any human correlates? This is actually a study involving the National Cancer Institute in the US, prospective cohort study examining green tea and colon cancer risk. Again, an association, but this is now in 69,710 women. This is a huge study. And what they found is drinking on average 2 to 3 cups of tea a day reduced the risk associatively by 57%, which is huge. So one question that we're beginning to ask is, well, how would you actually manipulate the food? So many of you will go to a restaurant and you'll order tea and they'll give you a tea bag. Well, how do you actually get the catechins from the tea? If you just drop the tea bag and let it to settle to the bottom, there will be the catechins will dissolve out. And we wanted to quantify that, so when you actually shake the tea bag, you kind of dunk the tea bag, as some people do, you get more catechins coming out. So manipulating the food and how it's actually handled can actually impact how much of the bioactive is actually consumed. So this is really some interesting ideas. Now, the other thing about tea is that tea influences the microbiota. So the tea we drink also influences the gut, and the bacteria in the gut helps the gut change its gene activity, which then can systemically influence the vasculature as well. So again, this interconnectivity is very complicated, and we're just beginning to be able to study how these go. So I've shown you many different foods. I can't talk about them all, but I'll just finish by talking about an area that we're working on now called the Health Expedition. This is really a new area for us, a new initiative, a global initiative, and very interested in developing collaborations where we're looking at the great expeditions of the 19th and 20th century in the Amazon and the jungles and mountains and in the South Pole. And we think that the big expedition now in the 21st century is inside our body in health defense and looking at how what we do in our lives, including diet, affects that. So we're beginning to look at things like differential activity among wines, so same terroir, same grower, different grapes, and to see in the same vintage what the activity of anti-angiogenesis is. So you can see in one research, we found 3 types of grapes that actually generate the most anti-angiogenic activity. We also looked at tomatoes. Tomatoes have lycopene and other carotenoids, including beta-cryptozanthin and others, that we know can influence prostate cancer. Are all tomatoes the same? No. Actually, we found that the San Marzano tomato has the highest potency, and San Marzano comes from the volcanic hills of Italy. The soil might influence it, the genetics of the plant may influence it, so we're interested in working with the agricultural world, particularly in Europe, where there's historically been great diversity of plant-based foods, and the pressures to try to do what we've done in America, which is to try to make all the foods the same, is a problem because we haven't yet fully understood how the different species are. This is the fall. We're in September in France. You go to the market in Paris, you'll see many different choices of apples. Which one might you take for cooking? Well, I leave it to the chefs. For those of us here, which apple a day might keep the doctor away for cancer? Well, we've looked at this for angiogenesis, and you can see if you look at comparative activity, the Renata apple, or Renette in French, is one of the most potent, but so is the Granny Smith and Red Delicious. And so choosing the varietal can actually have an influence. We've also begun studying in our health expedition the anatomy of a plant. So if you go to buy a frozen bag of broccoli, you'll see it's mostly the florets, and many people who buy broccoli from the market will chop off the stem and cook and steam or saute the florets, which are delicious. We wanted to ask whether or not the florets and the stem had differential activity. And when we did this, we found, in fact, that the potency of the broccoli stem had twice the amount of anti-angiogenic activity as the florets themselves. We also found this with carrots, that the green tops of the carrot was twice as potent as the orange taproot. Now, just to close, this is, how do we measure this? We are really interested in taking the tools of molecular and precision medicine that are being used in oncology today to guide our research on the connections between dietary health and cancer outcomes. So we can take a tumor, we can subject it to full genotyping, transcriptome, proteomics. We can begin to take a look at, match that with artificial intelligence to look at FDA-approved drugs or clinical trials. We think in the future dietary factors. And then there's even liquid biopsies which involve blood over time, easily obtained, to begin looking at circulating tumor DNA, exosomes, immune markers, and many other as well. So the unanswered questions that lay ahead in terms of dose and scheduling, biomarkers, augmentation of medical pharmacological treatment, and then how do you actually maintain balance, because we don't want to impair angiogenesis altogether. We want to keep normal balance, and I don't have time to show you, but there are other foods that can recruit angiogenic stem cells from the bone marrow to affect wound healing as well. So I'll just close with this quote from the Nobel laureate Albert Szent-Györgyi, who once said that discovery consists of seeing what everybody has seen and thinking what no one has thought. And I think what we're gathered here to do today is to invite, as colleagues, each other to view diet as something that everybody knows and presenting some data that might be new to each one. So I hope I've given you the beginning of some new information. Thank you.